Today, I'm about to speak here about company registration extensions. So it was partially already touched by Roland in the previous in the previous talk, and to give you a little bit of the Haystack project or Haystack story, which is something that we are also doing in ChemAxon. But first of all, I would like to actually clarify something because there are a lot of confusions regarding that. On my badge, you see consultancy, which means that actually we are part of ChemAxon. So we are working for ChemAxon. We are the so-called professional services of ChemAxon. We call ourselves consultancy just because we couldn't come up with any better name for that. If you have an idea, actually, how can we make this totally clear for everybody, please come to me and give me some ideas. That would be very welcome. So actually, we are a team of professionals who are having very different kind of skill sets. Some of us are biologists, some of us are chemists by major, some of us are chemistry engineers, some of our hardcore coders, and some of us, like for example, Brock, he just spent like 40 years in in this domain, I mean, the pharmaceutical domain. So we are the ones actually who wants to tackle a lot of problems. We are a little bit different than a typical ChemAxon product team because we're not a product team. We are rather something like a service team, and our main aim is actually to take care of these 4 or even more kind of things that usually we experience when we talk to you, and it is beyond what the off-the-shelf product can deliver you. So we do help you with designing some solutions, with some kind of architectural problems. We help you with integrating ChemAxon or not only ChemAxon services into your already existing environment. We do migrations, which is not limited to data migration. That's very typical to have data migrations, but we do workflow and other kind of migrations to whatever you can imagine here. And of course, customizations and extensions mainly to our products. So let me talk to you about the extensions part here, and let me just focus on one extension point that the company registration system has, which is the so-called workflow steps. So probably that doesn't come as a surprise to you that the registration itself is the workflow composed of different workflow steps, and this is highly customizable in our company registration system. Actually, you can edit these ones, and you can even add custom workflow steps into these ones. And today, I want only to talk about these extension points. So let me give you some examples. What can you handle with this very simple point? For example, the custom ID generator problem that Roland already mentioned partially, which is very easy. For those ones actually who are not in company registration system that much, assigning a unique corporate ID to a structure is something very important. It's probably one of the main values of a company registration system in a corporate environment. So you can have it for a single component, something like this one, but you can also make it a little bit dependent on the structure that you're registering. For example, if you have a solvent added to the single compound, then you can like introduce an extra S there, or if you have a polymer, then you can go on with this analogy and the mixture and the formulation and so on and so forth for no structure. This is sometimes very, very useful for the customers, and it's easy to implement using a custom workflow step. The next one is the custom field validator. So those ones who actually know company registration a little bit, they know that actually if you can put some validation on different fields like this one, and if you miss to comply with that validation, then it will just highlight it with red, or the system will notify you that, hey, there's something wrong with that. But what is the case when you want to make the validation dependent of a value of an already existing field? Like in this case, that the submitter, so the chemist itself, would not have access to all kind of lab notebook reference IDs, but some defined ones. This could be also handled by a custom workflow step in a very easy fashion. Actually, we have a dictionary service in our system, which would do nothing else but call out for a REST web service endpoint and say that Alice is allowed to register some kind of LNB lab notebook number, which has a different kind of, or a given kind of, validation. And that comes from a REST service. So that could be verified during registration. Another a little bit more generic validation is when you insert a structure, and based on the structure you want to have some kind of validation on your additional data. Like in this case, you have a stereogenic sander here on this molecule, and of course then you would expect the customer or the chemist to introduce something here at the stereochemistry as an additional information. That is also very easy to implement with custom workflow steps. Another one is that, however, during the registration, if you have salts and you assign to the salts some names, with a single registration, it's pretty easy to add salts to the to be registered compound. You just try to type here, and you have a lot of options to select from. That is unfortunately not true for the bulk upload. For the bulk upload, you would need to use the technical identifier of the, or the identifying number of the salt to be able to access this functionality. So what we did is very simple. We had an agreement with the customer that in the SD file that he's going to upload us, he's going to specify the salt name separately, like HCl, and we are going to map this one actually to the one that we have in our salt solvent database. So that way it is easy to register these ones too. Something which is very similar to this one is, again, when you have a stereogenic sander, you might want, or several times, people would love to actually make some notes. This is what we kind of call chemically significant text. Like this is a racemic mixture here on the page. And that is cool, again, when you're registering by hand, you have this possibility to specify this one, but it's a little bit more difficult to do it with bulk upload. Again, the solution is the same. If you specify this one in the SD file, and it's a totally valid SD file, by the way, we can identify this line and register the compounds as you would expect it to. So with this one, actually, I wanted to show you some glimpses of what are the possibilities only by introducing extra workflow steps or custom workflow steps into compound registration system. You can imagine that there is really a plethora of possibilities here. So the next thing that I would love to talk about is the Haystack project. And again, I'm going to start with an explanation. This is a project and it's not a product at the moment. We would love to have it as a product, and for that one, actually, we need a lot of help from you. And I'm going to just point out in what cases. Here, the idea is that seeing your systems and talking to you a lot, we realized that you have so many different kinds of databases in your system, which are piled up on each other in a very structured way, and sometimes there is not such a structured way. I mean here different kinds of databases on which you would love to search, and you have some important information in it. These piles are sometimes very distant from each other, even geographically, even digitally. They're sometimes packed, so you cannot just access them very easily. Or actually somebody's already using it somehow and already modified the data somehow, so you cannot just use it as it is. And with this one, actually, you can get up into this situation that you don't really know what to do. You have a lot of data, but you just cannot access them simply. So leaving the analogy, which might be funny, let's go back to this circle that you presumably know very well. So in the design phase, you have a lot of data coming from assay, or you're using a lot of data coming from assays, from different kind of corporate internal databases, or IP databases, being that some public database or your internal or something that you can access or you pay for. When you got to the stage that you want to make the compound, then you got, of course, to those public or vendor databases that you can buy maybe that molecule from, or if the molecule is not as accessible, then of course you would go for the building blocks to have that molecule synthesized, which is there. These databases are typically at the order of tens of millions or even hundreds of millions if you think about NME-real, for example. When you go to the test phase, you have a lot of similar assay data that are just stored next to each other. And when you analyze, of course, you can have SAR, catalog, scaffold hopping, and other kinds of databases with different kind of juggling. produced with this different kind of juggling. So actually, all this data is basically somehow scattered in the in your system, and that is what our proposition would be, to just unlock you the possibility to quickly explore your chemical space, whatever is there, to be able to search it and index an arbitrary amount of data, which is at least halfly created or somehow curated. And when we were also thinking about this and talking to you, we kind of identified two typical workflows. We are talking about huge databases here, right? So, I don't know, hundreds of millions of data, of rows of records. So one of the possibilities that you have a workflow usage, which means that actually you want to retrieve all these structures and all the records. You know exactly what you're asking for, so you know the query, what you have to trigger, but you are also interested in the whole result set. In this case, we have very good workflow steps, workflow handling products on the market. So actually, that's not a big deal. You just put together your workflow, you fire it, and you wait till morning till it gets the result set. The other workflow is when it's an interactive thing. So you're not exactly sure what you're looking for, and the whole processing is made by a human. And a human would not be, or not likely, will be able to process millions of results. Then it makes no sense, actually. So here we would love to concentrate and focus on those cases where there is just, I don't know, hundreds of results coming back, but those are the most relevant ones to your question. For this one, to tackle this case, and that's what Haystack is about, we needed some technological knowledge or prerequisites, so to say. The first one is the relevance search. You heard about that from Wolfie yesterday. So if you would do the search with these hydroxycoumarine, for example, in this case, this is what you would get back as a result set for the first few hits. It's correct, so it's a substructure of it, but they're rather big and they're rather distant from what your question originally was. So what if we could sort these ones and give you back just the relevant ones, those ones which are pretty similar to your original query? So that was one of the prerequisites that we needed for Haystack. The other one was actually that, well, it would be very nice if we would be searching on a huge database, and our results could come back just very quickly, as you can see it here. This is what we call the hit as you draw. Here the trick is that it's not the whole result set, but it's the first, I don't know, 100, tens of hundreds, thousands maybe. So you can limit that one. So with this one, actually we tried somehow to squeeze together a lot of databases that we know about that you're using, which is your corporate database. For example, this is usually at the order of a few millions or tens of millions. We have some screening libraries. These are typically around millions, if we know it well. You have Campbell, so public databases of Binding DB for biological data, for example, or Sure Campbell if it's about a patent. We are already at like 20 millions of records. And then comes the bigger ones, the e-molecules, other public vendor databases, PubChem, for example, Molport, or of course the biggest one is the Anamin Real, which is just right now got to 720 millions of records. So roughly this is what we think is that this is your chemical space that you are working in, and you would need to tackle these ones. So how did we do, and what did we do with these ones? Well, we literally squeezed them together, and we put a nice application layer on top of it to design our Marvin Live application, just to make it tangible for you what the backend is capable of. So we are using it just for visualization purposes. So this is how you can interact at this moment with this application. But of course, we would like to make you available this to be accessed by different kind of technologies or interfaces. And we had no better idea to put here something like stuff. We don't know exactly what that API would look like at the moment, and you're going to understand that one in an instant. Why is that? So what is the technology behind this? The first one is that we have these databases which are very relational kind of databases. So actually next to the structure, you have a lot of additional information which might be of your interest. So we figured out that actually we don't want to lose any of this data, and then your need for these data is very different. Usually, so some people are looking for different kind of aspects, the other for other kind of aspects. So what we did here is that, okay, let's just sort them by uniqueness, so deduplicate these databases. We know from their way of composing them that they are sometimes overlapping to each other, so uniqueness filter would be good here. We got to 120 million unique structures here. And of course, we use PostgreSQL and Postgre cartridge, ChemAxon technology to search on these ones. They're nice relational databases. Why not? We could index it, so that's not a problem. It works very fine. There is another thing that we thought of, is the enemy real, the beast, the big one, the 700 millions. This one is pretty flat structure. So actually, it has only the structures and an ID. So for this one, actually, we thought out that actually ChemAxon microservices is a very good tool to handle this one, as you can load it and search it quite extensively. In those cases, when you would actually love to use different kind of similarity, different kind of similarity metrics or different kind of fingerprints to get to the similarity searches, we implemented MetFast for that one. MetFast is really flexible. You can define your own fingerprints there, and of course, you can also give the totally custom metrics for the fingerprint that you define. So we think it's a pretty nice thing. And above of these, we just put for visualization purposes the Marvin Live frontend to really make it tangible somehow for you that this is the backend. So my next slide is saying demo. So I want to demo for you how fast that is and how does that look like. So this is what we have right now. And let me just go with this one. So what you can see here, and I hope it's visible for the back of the room too, is that basically as I'm drawing the structures, you have the searches done in this 120 million of structures. These are just substructure searches. And of course, we had no better idea than just put here the link for PubChem, for example. So you have it drives you to the PubChem web page. And here you have a distribution of the results by databases. So you can see that 35 was from Schur-Campbell, some from eMolecules. And if you're worried about the numbers that do not add up to the limit that we set to 100, is because different databases can have overlap. So the same structure can be present in different databases. So let me go a little bit further with this one and show you the similarity. For this one, we used, for example, ECFP4 fingerprints, which is pretty standard, I believe, on the market to use. You still have the substructure searches, substructures, and you have also the similarities, and of course the result distribution for this one. And again, let me just show you the speed of the system, which is what you just see here. So let's go for the big one. What if we can put all these things together? So you would have the project Haystack, which is the Postgre database with all the relational data in it. You have the fingerprints, and you also have the enemy real with all the compounds in it. So let me just, in this case, to delete some. And roughly this is the speed that you're experiencing on hundreds of millions of structures for speed. And with this one, of course, the links do work, hopefully. Yep. And with this one, I would love to conclude my presentation, and I'm really open for any questions, discussions. Please find me. If it's a little bit longer, please find me in the breaks. We'd be really happy to learn about your use cases you could use this technology for. So thank you very much.